third session of the day, and it is my pleasure to welcome our next eminent personality, Dr. Satish Patel. On behalf of SSR College of Pharmacy, I take the honor to introduce Dr. Satish Patel. Sir has completed his post-graduation and PhD in the subject of veterinary pharmacology and toxicology science from Anand Agricultural University, Gujarat. He has more than 22 years of vast experience in this field. During his career, he is involved in establishing many animal facilities at different organizations. Amongst few are GLP and American Association for Accreditation of Laboratory Animal Care qualification. He has also developed preclinical disease models, including transgenic animals by different tactics, which are useful for research in different therapeutic areas like diabetes, inflammation, arthritis, dyslipidemia, cancer, etc. He has vast experience for maintaining and conducting experiments on different rodents and non-rodent species, including dog and non-human primates. He has worked for B.V. Patel Pharmaceutical Education Research Development Care, that is PERT, Hester Bioscience Limited, Cadila Pharmaceutical Private Limited at different capacities. He has also worked for Nova Pharma Canada, which is now known as Teva, a largest generic company in Canada. He has published more than 25 publications in national and international journals. He has also traveled few countries like USA, Canada, Netherlands, Spain, Italy, Germany, Singapore, Malaysia for academic as well as research purpose. He is also associated with society like Laboratory Animal Science Association, Indian Society of Veterinary Pharmacology and Toxicology, and Gujarat Veterinary Association. He is Joint Secretary at Gujarat Veterinary Association and ex-member of Executive Committee of Gujarat Veterinary Council. He is also an executive member of Laboratory Animal Scientists Association and involved in many ethical activities and also a member of IAC in different institutes. He has delivered many guest lectures to different pharmacy colleges as well as presented seminars and lead papers in workshops and conferences. He also organizes and gives training to animal scientists and experimentalists. Currently, he is serving as General Manager and in charge Animal Research Facilities at Zydus Research Center, Ahmedabad, Gujarat. Today, Sir will deliver a session on test systems in regards to GLP. With this, I request Sir to kindly take the charge of session. Sir, please.
Yeah, thank you, Dr. Niyati, for a kind words and introduction. I'm especially thankful to Dr. Sanjeev Acharya for giving me this opportunity and SSR Trust for organizing this seminar to update the knowledge of faculty. And this is really wonderful. I could attend in inauguration function where also many dignitary of national authority in GLP have been participated and blessed this occasion. So I'm really thankful for giving me this opportunity to deliver the lecture, especially on test system in GLP. Thank you. And also pardon me for rescheduling the session for half an hour delay due to some other occupancy. And I thankful to management for allowing me to take that privilege for 30 minutes, and sorry to participant for same. So now going to the topic, you are listening since 2 days about the GLP. So I need not to go more detail on the GLP, but going to the test system in GLP, I would like to introduce little about GLP background, history, and why the test system is important and where it's located in overall GLP compliance. So, and why the GLP is required? Which study required GLP? I mean, so there are the evidence that that product are safe. The research institute or pharmaceutical company like us those who are marketing the product in the field and for a human treatment. So they have to they work as a sponsor and they conduct the study for proving the safety of their products. So there are many other industry, food and color additives, animal food additives, animal drugs, human drugs and biological products, medical devices for human use, some electronic products, where those who are being used by the human directly or indirectly, to prove that safety level of those components or things required to pass through the safety or animal study. And now those study, if it is being conducted in the regulated environment, so that reproducibility as well as comparison of the study results can be done from, if it is being conducted at India or it is being conducted at Europe or USA, anywhere. But the basic criteria or procedure of conducting the experiment would be harmonized, then the result are expected to be a similar kind, and the drug or a product or a food additive or any cosmetics that can be evaluated and given to the human use with safety margin. So the mainly the non-clinical laboratory study that must comply with the FDA and GLP regulation, which includes the toxicology profile. So toxicology profile, no, there is NOEL where there is no observed abnormal effect, risk of clinical study involving human or animals, potential teratogenic, carcinogenic, and other adverse effects. These are the study conducted to evaluate this effect, safe level of use. So what would be the safe level if you are using any drug? Paracetamol, we very regularly we are consumed for fever or any analgesic, if you see. So what would be the safest level? So you everybody knows who are studying the pharmacy LD50 and how they've been derived. So you might be aware about this. So the non-clinical laboratory study that must comply with the FDA GLP regulation, which are going to further human consumption. So, and there are few studies which may not require to conduct under the GLP condition. There is a basic research, discovery, screening of compound. So, at present, the pandemic COVID is going on. So, those study before going to the market, if we are developing the vaccine or any other institute involved in the any drug developed for the COVID treatment. So, those products before going to market, they have to pass through the regulatory experiment which required to submit to the regulatory body, and those experiments must be conducted under the GLP environment. However, the first research will definitely require to conduct as a basic research where you may not be able to follow all the GLP norms and regulation. So once you find out the lead candidate and you think it is efficacious and you can develop it further, those molecules can be take up for further development, and the drug development process must be do through for preclinical study. It should be done through GLP experiment only. So now coming to the introduction to the GLP, what is the GLP? So if you see the good laboratory practice, a quality system concerned with the organizational process, the condition under which non-clinical health and environmental safety study are planned, performed, monitored, recorded, archived, and reported. So this is the basic thing what you are doing under GLP. You will plan the experiment, you will perform the experiment under specific guidance, that is SOP or regulatory guidance, OECD guidance, ICH guidance, WHO guidance, and you will monitor as per recommended process. You will record each and everything during the study, and at the end of the study, you will prepare the report. And you will submit that report along with the raw data to the regulatory authority. However, you will archive the report, including along with the raw data, to the archival, and that record you'll have to maintain till at present it's a 9 year, I think, as per the regulatory or GLP requirement. So, drug company where laboratory or GLP, they conduct the experiment under GLP condition, then they submit to the government, and after evaluating the hazard assessment, the regulatory body approve the drug. So it's a non-clinical health and environmental safety studies, where non-clinical is not in hospital, that is a preclinical, what we are doing on the animal health for human being, and environment safety study that is being conducted on GLP. If you look at the history, there are few incidents, very fatal incidents, happened in the past in USA before 1970. I think in 70s, the thalidomide example is well known. Many pharmacologists might be knowing about that. So that GLP before, so we were at the history of GLP when how it evolved, and what happened in the past, which enforced the people in this industry to make one system, harmonized system, so they can conduct the experiment in harmonized way to compare the result and reproduce the same. Yes. Okay. So first, before GLP came in picture to the US FDA, the first introduced by the New Zealand government in 1972, and whereas in US it introduced in 1976. So mainly it came up just because of the industrial biotest lab who have submitted the wrong data to USFDA, and due to that, many casualty happened to the public by consuming such drug which has been tested on animal and approved by regulatory authority, though they were have safety issue, but that was not reported, and drug got approved by regulatory body. So the case, there are very well-known example of thalidomide, sulfanilamide, where after consuming thalidomide drug, the pregnant lady who delivered the kids having teratogenic effect, and after evaluating the case, government and US FDA understand that we need to have some system which can monitor the study and result, which can be comparable and safe. So that's how GLP came in picture, and it regularized and introduced officially by 21 CFR Part 58 on December 22, 1978. So this was happening in USA. So, and after that, India was for a long time was a member of OECD. So the organization named in 1981, after 1979, the OECD came in picture. That is Organization for Economic Cooperation and Development. That is which is produced or made on the base of GLP principle, and as per international standard designed by the US FDA. And right now there are many countries other than the US and Canada, those who are not following the 21 CFR Part 58, they are following the OECD guideline. And OECD guideline is acceptable in US when you are conducting any experiment in India under OECD GLP, that is acceptable to the US government. And we being a pharmaceutical, we also follow the OECD GLP principles. So this is US and Canada is US FDA, that is 21 CFR. Other than the US FDA, Canada, Australia, New Zealand, India, Europe, all are following OECD GLP. So that I already noted, the objective of GLP, that is GLP makes sure that the data submitted are true reflection of the results that are obtained during the study. GLP also makes sure that not to indulge in any fraud activity by lab, and promote international acceptance of taste. Earlier, as a history, we have been heard lot of many times that whatever experimentation have been conducted in India, there may not have any quality. Even research paper also, good impact journal, always doubting on research data submitted from India. But when it has been raised through the GLP monitor lab, then they don't have any chance to doubt on those data, and they readily accept and publish those data. So this is what impact we feel once you have a GLP certification. So there are few principles through which the GLP being monitored. So that these are principal are test facility organization personnel. There are a TFM, the study director, and many other personnel. They have a defined role to perform in the study. There is a QA, learned QA people who monitor the study through conduct from concept till final report, facility, apparatus, material reagent, test system, test reference item, performance of the study, how will you perform, reporting of the study. There is standard format and guideline for conducting all these activity listed under the GLP principles. So, hence, test systems is connected to the facility. I have kept one or two slides regarding facility before we go to the test system of today's topic. So, what type of facility required for conducting the GLP study? So that there should be a sufficient number of room or area that assure the isolation of test system and individual project. There are maybe many study going on simultaneously. If we are talking about us or any lab having test facility should must be well equipped. You must have to have a isolation area for animal, for a project, for a sex or a species. So you cannot keep everything together in a single space. So you must have a sufficient number of rooms or area for isolating the specific activity. There should be a storage area needed for a supply equipment. You must have a case storage area, feed storage area, bedding material storage area. So the in case of animal facility, the support area that is other than animal room are always 2 to 3 times more required, 2 to 3 times more than the animal room. So in normally in an institute, research institute or academic institute, whenever they design the animal room or animal house for a particular GLP study or permission in the pharmacology, so as a part of academic approval, so in that case, they made only one or two room, put one air condition unit and showing the regulatory authority. But nowadays, things have improved a lot due to the CPCSCA in India. ALAC, as Madam said, the ALAC accreditation is a very critical accreditation for an animal welfare point. So now this level of animal housing has been improved. So the storage space especially is very critical and important when you want to maintain the quality animals. So areas should be available for diagnosis, treatment, control of disease in order to ensure that there is no unacceptable degree of deterioration of the taste system. So when if you don't have any enough space to isolate the diseased animal or a affected animal that may contaminate or infect other um, animals which are housed together, and that may impact the study outcome. So there should be enough space in case of facility. That's why I kept the slide. You have to have a dedicated archive facility, which where you can store all the reports, sample, test item, specimen. So this archive facility should be also designed in such a way that environmental condition can maintain the stored specimen or a document for a longer time without deteriorating its quality. So as per a present recommendation, 9 year minimum they can things can be stored without deteriorating their quality. There is a waste disposal. There's hence you are handling toxic, probable toxic compound It may not be a toxic in all cases, but when you are testing those compound or on animals, so the waste disposal become very critical in that case. So you must have to have a way not to jeopardize the integrity of the study and dispose the thing handling in such a manner that it should not harm to the handler as well it should not harm to the animals. So waste disposal is very critical part of animal experimentation, and at present there are defined guidelines. You have to incinerate the carcasses once the experiment is terminated and animal are sacrificed. Earlier, people used to burrow the animals in their campus, but now it's not allowed. So you have to have, or there are agency are also available who can do this activity on behalf of you, and they can collect animal carcasses from your facility, including this biohazard haste, and they incinerated their facility. So these are the about the taste facility because it's very connected to have a better study outcome. You have to maintain the taste system in such a manner that study results are not impacted, and to manage and monitor study in a good way, you have to have a good facility which can accommodate the required animals as well as support all the animal related activities. So now coming to the taste system, the core topic of today my presentation, and is important in OECD or a GLP. So if you see the taste system is not an only animals, it's a biological, chemical or a physical system or a combination of it. That may be an animal, that may be a plant, that may be a bacteria, organs or a cell, and it also can be an analytical equipment also a part of the taste system. So before going to the in animals or in vivo system, there is a GLP taste system which defined as an in vitro system. So for better understanding, I have divided this taste system in two parts, that is in vitro system and in vivo system. So in vitro studies, that those studies which are don't use the whole animal or a plant are defined as a in vitro studies, and that involve the use of subcellular fraction, microorganism, extract from animals or a plant and isolated organs. So in most cases, the studies are short duration, and the OECD consensus document the application of the GLP principle to short study is applicable in vitro GLP study conduct. So yeah, many faculty might have done their work on in vitro system during either in cell culture or a plant cell or any other in vitro system during their academic career. So if you see the in vitro method have become more common in the field of drug and safety testing. At present, there are many agencies in the India as well as abroad. You might have heard about the PETA, other animal welfare activists, those who are preventing or opposing the use of animals for experimentation, and they have that is very strong group, and they are working a lot to prevent close the animal experimentation, and they are promoting 3 R's. That should not be animal uses according to them, but the regulatory body or a scientific faculty, those who are advocating that wherever required, we must have to do animal experimentation. So, in this scenario, as far as possible, we must use the in vitro system unless and there is no other way. Those cases only you have to use the animal. So, in vitro system is very critical nowadays, and as far as possible, you first try to use the in vitro system. So in vitro cell or cell culture based test system are routinely used by industries and regulatory body in toxicity testing as well as safety assessment and risk evaluation. You might be aware about the pyrogen testing earlier when we were studying, even after our carrier joining, people used to do pyrogen testing in rabbit, and but now it has been replaced by the in vitro test lal. So there are many example which earlier used to be done in animals, but now has been replaced by the in vitro systems. So that's how in vitro system is equally important in GLP as well as in animal experimentation. And in case of in vitro system, to claim GLP compliance, in vitro method must adhere to the principle of OECD, that is a planning, performance, recording, reporting, monitoring, and archiving. That what we so all in vitro experiment should be performed according to the GLP experiment under GLP condition. However, there are some limitation of in vitro system. If you see that you cannot conduct the toxicity study, especially the new chemical compound study, where you never know how they will act on the off-target receptor in the animal body, so that you cannot test such compound in vitro system. But you, in that those cases, you have to have an animal as per presently available scientific information. And one of the difficulties with the in vitro method is that they usually require trial substance in addition to the test system. So you have to have a comparison of a reference compound when you are evaluating your compound. So substance may be used to demonstrate the appropriateness of the test condition. This means that standard GLP definition of the test item and reference substance doesn't strictly apply in this case. So this is the limitation of the in vitro system. However, if you see the OECD document on in vitro study, that always mention that other trial material should be considered as a reference material for GLP purpose. But it may not be possible or appropriate to characterize them in usual manner. So when you are comparing the reference standard, so you cannot always characterize those reference standard. And in addition to that, you may require a specific facility to conduct the in vitro testing. So if you are doing the cell culture based assay, then you have to have a defined environment. You have to have an incubator, or as you said, the P2, P3, P4 type facility, a biosafety level facility. In and concern are center on the viability and the quality of test system and the level of potential contamination. So there would be always chances of minor contamination, which may disqualify the test. So in vitro system, you have to have a dedicated and desired facility which require basic infrastructure to maintain the biosafety level, as well as other environment required to for such a test which are being conducted in those facilities. So, hence, the facility and equipment used in vitro study are likely to be sensitive to contamination or may be critical to the viability of the test system. It is obvious that related maintenance and calibration process must be fully controlled and regularly performed. So, this is a very critical component of in vitro system. So, whatever equipment or instrument you are using for evaluating such in vitro assay, those instruments shall be required to calibrate or validate at the regular frequency. Normally, it's biannually or annually once, and it has to comply the specification. If it is not comply within a specification, that instrument or equipment required to repair or replace, and you cannot use until it comply the desired specification. So frequent monitoring of those equipment is also important, and calibration validation report you have to maintain and archive it annually. And in addition to that, environmental monitoring is also important, where you are performing the test. So, in vitro lab environment is very critical, and you have to monitor those environment for that quality or as well as viability of the test system. And it is important to determine before the tests are performed which monitoring activity are essential and the frequency at which these will be performed. So this you have to define this thing under GLP before you initiate any experiment. And you have to record those activity accordingly, environmental data, and maintain those records. So whenever you are after completion of the experiment, when you are evaluating the results, so you can analyze the impact of any variation happen in environment or instrument and how it impacted the outcome of the result. So this will be helpful for interpreting the in vitro system. Now the main test system in GLP, that is the in vivo, where the animals are being used. So test system is animals. You might be, all the pharmacy student and faculty use animals at least once in their career during bachelor or master academic career. So, but now the GLP compliance and compliance with animal welfare. The whenever you are using the animals, and the one thing you must understand and remember that animal how to maintain, treat them. So that is there is a defined guideline for animal care and use. That what we follow here in our institute. So it's an international defined guideline. The guide is the name is the guide. So it should be complies the animal welfare legislation. So all the rules and regulation prevailing in concerned area, that's in our case is India, and there is a CPCSE. So before GLP, you have to have, comply the welfare legislation of animal. And then after complying the welfare legislation, you are free to conduct the experiment. And experiment you may conduct as per GLP, or it may be non-GLP. If you are conducting as per GLP, then it must be comply with the GLP requirements. So what is the GLP requirement for the test system? And that is for animals. So that scientists must match the quantity and quality of animal to research requirement. So, whenever you are planning any GLP experiment, though you must be very clear with how many animals you may require, what type of quality you will be required, the what weight range you may be required, what age would be required, which sex you will be using. So, when you are planning to conduct any GLP experiment, being a study director of that experiment, you must know the what is the phenotype or a genotype of animal selected, or you are going to use, suppose you are going to use the Vistar or SD. You might be aware about the rat, very vast people are using very commonly in all the colleges. So the Vistar rat and Swiss albino mice, now it's become a ICR, because Swiss albino doesn't have any proven pedigree record. So now internationally, SAM is not acceptable, but ICR is International Cancer Research Institute original strain of mice is being globally accepted for a toxicity study, and throughout the globe people are using the ICR mice for a toxicity study. So whenever you are planning any experiment on animals, so test system, you must be aware about the phenotype or genotype, what is sex, and who are going to, from where you are going to procure those animals. The supplier must uh, have a quality animals, because you cannot procure animal from any breeder or anywhere in the globe. The wherever whichever institute or breeder supplying animal, they must have a pedigree record, good quality control record, disease monitoring record. Animal should be healthy. So all the records should be available with the supplier, and you being an experimentalist or a study director, you must select such supplier which having all the quality data required as per the GLP principle. And secondly, you must also be aware that why you are selecting suppose Pragdol here, being a study director, you selected a Sprague Dawley instead of a Vista. So you must be clear that why you have selected that test system, and that must be mentioned in the study plan. Because the reason for selecting those strain or a species, there are many study we have to conduct in the rodent. Then as per regulatory requirement, we have to conduct two study in the rodent and one in non-rodent. Non-rodent, you see that there may be a dog or a primate. So that is the regulatory requirement. So you have to have a reason why you have selected a dog, because once you are selecting a higher sentinel animal, that is a dog or primate, the animal welfare or a CPCSA or a regulatory body will not easily approve those study for conducting experiment in higher sentinel animals. So you must be clear, and you have a scientific as well as a logical justification why you are still using that species or a strain for such experiment. And then you also have, regarding the animal, specifically to the animal, you have to have a very clear understanding and record as well as data available for regarding the species characterization or strain characterization, what health status of animal at whenever you are receiving the animals. During the study also you must monitor the health or clinical status of animal, the supplier quality, how they are maintaining the breeders, what frequency they are evaluating the health status of animal, how they are what they are feeding, what their type of treatment or precaution they are taking, how what type of environment they are maintaining the animals. Yeah, and the same environment is going to be maintained at your facility or it is a different. So these thing you must be aware, and you have to have a ready data about that. What are the background data of a selected strain or species? So unless you are aware about the background data, you may not
Okay.
be. So you must have a background data. Unless you have a background data, you cannot interpret the study outcome and compare the or efficacy or a toxic level of the compound unless you have a background data or a basic data of those animals, and the separation of any, whether the specific strain or species have been housed separately or in with other animals. And for your knowledge, there are, if you cannot house the animals in a single room of two species. So if and normally people are housing rat and mice together, there is a pheromone effect. So hence you are conducting the experiment. It is not advisable to keep two species in a single room. How far now the new technology develop, and that is the IBC. You might be aware about the individual ventilated caging system, where the exhaust air directly goes to the environment and it doesn't contaminate the animal room environment. So whatever pheromonic effect may not harm the other species housed in the similar room. But when you are housing animals under the conventional system, you must have a separation of the species wise animals. So whenever you are conducting the GLP study, so in case of husbandry, so separation is a part of husbandry. So you must follow the national regulation, what they have recommended. You might be aware about the CPCSA guideline as per our national regulation is concerned, where they have recommended to maintain the animal between 18 to 29 degrees Celsius temperature. But ideally, the rodent, as far as rodent is concerned, you should maintain it 23 plus or minus 2 degrees Celsius temperature. But considering this guideline drafted in back 60s, 70s, that time, considering the Indian scenario, they have kept such a wide range. But ideally, when you are submitting data internationally, you must conduct experiment in internationally acceptable environment. So, for husbandry, you must check on the room cleaning. So, whether rooms are being cleaned every day, twice a day, or thrice a day. or a weekly, depending on the activity involved in the experiment. Cage or rake are being cleaned routinely or not, at what frequently they are changing, because if you are conducting any experiment having the diabetic effect, frequent urination is very common. So in those cases, you have to have a higher change the cases at higher frequency. If you are not changing the cases at higher frequency, then it may contaminate animals, or in may reduce the immunity of animal. The animal cases will be dirty, and ultimately your experiment result will be impacted. So feed and water quality, what type of feed you are providing to the animals, whether feed has been tested for the contaminant analysis, proximate analysis or not, does it have any pesticide content? If yes, at what level it is under the recommended level of the internationally accepted guidance, or a water is contaminated, or you can provide the UV treated water or RO filter water, where the water should not contain any E. coli or Salmonella, which may harm to the animals. So these are the husbandry criteria or a specification you must be aware about, and you have to ensure whenever you are conducting the GLP study, especially in the rodent. The environmental parameter, as I explained, the temperature, humidity is the 30 to 70%, whereas in India you may find higher humidity in case of monsoon season, very lower in winter. So, however, you have to have some mechanism which can maintain the humidity between 30 to 70 as per our Indian guidance. That light, what illuminous or a lux level you are maintaining in the animal, that is, we have a defined guideline for maintaining the lux level in the animal. Air changes, the air changes of 12 to 15 air changes are required. So the complete air volume of the room need to be changed at every hour. 12 to 15 times that volume should be replaced by the pressure. So this is a guideline. So whenever you are designing the air handling unit or AHU system, you must take care of such specification. And then document, you have to check regarding these husbandry practices, whatever you are following, and it should be monitored regularly. So, and the process should be recorded. So people will not miss any activity like feeding, watering, because animal is not going to speak whether the feed is provided them or not. So yeah, you must have a document check on those. Whenever there is any deviation happen due to any reason, suppose in lockdown, people may not be able to comply all the activity or attend all the activity, or how we have suffered. So in those cases, how you have monitored, which are the critical component of the study which required to monitor daily without impacting the study outcomes. Such activity, how you have managed the remaining activity which may not have an direct impact, how you have managed that type of any deviation happen, you must have to record and report with the study outcome. And so, how you will assign the group animals to the different group? There is a low level, low, mid, high dose toxicity group, recovery group. So that assignment of a group you must have to define in the protocol. Whenever you are preparing the study plan, you must define the uh, uh, procedure that how many animals is a 5, 6, 3, 10, how many animal you will take for a single group, low dose level, mid dose level, high dose level, or a control group. So that has to be very clear. Also, you have to keep the data used for the assigning the why you have keep the 10 animal, not 5. So you have to have a logic and the data used to derive that figure of animals proposing under the protocol. Then you must have a location of a rack or a cage in the room. If you have many scientists are working in a single room, then in that case you have to have a location or a rack case documentation, whether it is in the first or a second or a last position in the right side of the room or whatever. So all the documentation, and if by chance anything that is a case rack or disqualification happen in test system or environment, then that must be recorded, and its impact must be analyzed and should be reported at the with the final study report. So the test system having a specific identification, which is very crucial. When you are conducting the study, how will you identify the animals? Which is a very critical part of the GLP study, because when in case of carcinogenicity study, you have to dose almost 15 to 1500 to 2000 animal every day. So whenever you are dosing the animal, you must ensure that you are dosing the right animal with right dose. So for that, the animal identification is very critical and important, and those animal should be acclimatized with the identification mark. Previously, people were using the picric acid, which is completely banned and not recommended at this time. People also use the permanent marker, that also I would not recommend to use. And then the tattooing or permanent ink marking, aims is also available. But right now, presently, very cheap micro RFID are available, and I would recommend to use those identification microchips for animal identification. Large animal individual mark or tattooing is also still acceptable, but at the latest advancement and considering animal welfare, the best is the microchip, that is RFID, and you have to implant that RFID subcutaneously in the flank or region, where you can easily detect by the reader, and you can see the animal identification and proper animal dose or handle, administer, bleed, or whatever data you are collecting, that would be linked to that animal number. Previously people used to have only clays label for a small animals, and individual unique identification by just marking a tail. Presently, many institute or laboratory are still following, but there may be always chances of misleading the animal number. So it is advisable to use RFID or a tattooing in like identification, and that animal ID should be mentioned in the all the data or recording form, and the ID must be checked regularly, because by chance animal being a live system, if that ID has been removed or spilled out from the animal, and then it would be difficult to catch, because it will mix in the bedding material and it will go in the waste disposal, and you will never realize unless you check it. So the ID, animal ID check is very important. Then animal acclimatization. So length and depend on the acclimatization is also important. In case of a large animal, you have to have a longer duration acclimatization. When if you are procuring animal from outside from other facility, then in those cases you have to give enough time to acclimatize the animal under your facility environment. And if the animal are being used from same facility, then you may reduce the acclimatization time. Example like institute like us, where we have a same facility, and within facility animal are breed and same animal will be using for experiment. In those cases, acclimatization time you can reduce. But when you are procuring animal from outside, you have to have a recommended acclimatization time. You have to check the health at specified time. When you are receiving the animal, at that time you have to check the health status of animal. After certain quarantine period, you have to again test the animal before you enroll them for a study. So whenever you are enrolling animal for a study, that time healthy animal only will be enrolled in the study. So you have to have this document of all the health record and approval for the study, enrolling those animal under study. So these are the different aspects you have to monitor while regarding the taste system under GLP. So when you have acclimatize, but before receive, uh, when you will acclimatize, you before acclimatization you will receive the animals. Then here if you are received from within a facility, there won't be any issue. But when you are receiving those animal from outside institute or from any other breeder, then you have to inspect a few things when animal arrived or you received those animal. So is for as per you ordered, is the health status is okay, any animal is ill or not good, feeling bad, not eating or drinking water, such animal you have to monitor very closely. Is there any transportation stress or animal has having any health issue? The sex of animal, whether the animal uh, is as per your order and as per required quantity of male and female order has been received or not, that you have to check. The number of delivered is as per order, the weight and age are as per your order or requirement, that you will have to check when you receive the animal. You have to record the receipt data. Any deviation from the specification, that what you have given animal specification, the protocol is as per the specification animal has been received, or is there any deviation that you have to mention in the record of receipt, and you have to check the against the protocol requirement, and after receipt you have to stock those animal in the clean room. So after receiving and quarantining, then you will also have to evaluate the supplier qualification. Yeah, and that is usually done by the scientist or a QA personnel. That is being assessed for the quality of animal being supplied by the supplier. And so supplier qualification is a part of any test system in GLP. So whenever you are receiving any animals for a GLP experiment, the supplier or of animal is should be qualified by your GLP authority, monitoring authority, that is your QA or a TFM, and same to the bedding and animal feed. So, where you are getting the animal feed or bedding material, those supplier also have should be qualified through your trace system or GLP system, and you also have to maintain the raw data for order form, supply invoice, transport certification for your record. Additionally, as mentioned earlier, for the environment study, GLP says study report must contain a description of all circumstances that may have affected the quality or integrity of the data. So, in short, if the a temperature or a humidity went beyond the recommended range, you must have to record those such deviation in your recording system and mention. And at the end of the study, you have to evaluate the impact of those deviation. So, in which circumstances the environmental condition has been deviated, that also you have to record. So these are the part of the case system importance. How will you monitor? From where you will bring after receiving? What will you do during the study? What type of things you will monitor? Environmental monitoring, then health monitoring, and at the end of the study, how will you terminate the test system? How will you dispose of? These are the critical factor of a taste system under GLP environment. So this is what I have just compiled the things for your review. That's are proper conditions should be established and maintained for a storage, housing, handling, and care of biological system in order to ensure the quality of the data. For any biological system, you have to for a test system you have to consider these are the few points, brief point. Newly received animal or plant system should be isolated until their health status has been evaluated. So you cannot directly mix the new arrival animal, newly received animal with your existing animal. You have to keep them isolated for a quarantine period till you do all the testing. Once you find it's complying as per your desired specification, then only you can take those animals for a normal study or housing in your facility. So and during the quarantine, you must observe the unusual mortality, morbidity, or the particular lot of animals should not use in the study. And if you find such morbidity or mortality or any infection, so such lot of animals you should not use in the any experiment. And such animals you can humanly sacrifice. And any system you find that are not healthy should not be used for experiment. So whenever you use any animals or a test system should be free of any disease or condition that might interfere with the purpose of the conduct of the study. So you must ensure that whenever you are using any animal, that should be free from any illness or infection. So test system that become diseased or injured during the course of the study. So you will take the animals in a healthy condition, but there is may a chance your study duration is longer. Then there are a chance that animal may fall ill or injured or anything can happen to the. So what will you do? So that time the test system become diseased or injured during the course of study should be isolated or treated if necessary to maintain the integrity of the study. Any diagnosis or treatment or any disease before or during the study should be recorded. So, if there is a human endpoint, you might be aware about that term. So, when in specifically a toxicity study, when animal having a severe toxic effect and animal is suffering a lot for that living, animal is not taking food or water, so in that cases, the study director must take a decision to humanly sacrifice those animal. So if it is having impact of a study outcome, even though you will have to sacrifice those animal and you have to record it, and you may exclude or include that data in the study according to the guideline or a scientific justification. So this is a very critical part of the experiment. So people are just crazy about their data that if they have selected 6 or 10 animals, that at the till end that animal number should be maintained. So there should not be any deviation in the statistical analysis, irrespective of animal health or animal suffering. But it is not likely the case. You have to have a must define the human endpoint, and whenever you find such a condition, you have to take a decision to remove the animal and humanly sacrifice. And eventually you can analyze the data by recording or removing that animal data or results. So records of source, date of arrival, arrival condition of test systems should be maintained. So these are the critical part. Just I am enlisted to give you a brief description about that. Biological systems should be acclimatized and test to the test environment for an adequate period before the first administration or application of the test or reference item. So that's what I explained in the acclimatization. All information needed to properly identify the test system should be appear on their housing or a container. Individual test systems that are to be removed from their housing or container during the conduct of the study should be a appropriate identification whenever possible. So that's what I. Whenever you are removing the animal, that must be having a proper record with its individual identification. During use, housing or container of a test system should be clean, sanitized, That is what periodically you have to change the cage, wash the cage, and sanitize the cage. Any material that come into contact with the test system should be free of contaminant at level that would interfere with the study. Bedding for animals should be changed as required by sound husbandry practices. Use of pest control agents should be documented. Test system used in the field study should be located so as to avoid interference in the study from spray, drip, or from past uses of the pesticide. So, whenever you are having a defined pest control system, you also have to take care of this. And at the end, I just it's additional to the test system, but very critical component of GLP study is the dosing. So whenever dosing animal orally, you have to take care of this part, because when your taste system is okay, your formulation is everything you have monitored, but your act of dosing is not proper, then there is many chances that your data will be not as desired, according to the GLP requirement. So whenever you are dosing the animal, you must take care for the animal are dosed group by group. So it should be from low level to higher level. You should not dose from the higher to lower level. So it is advisable to dose the animal from lower to higher. So that if by chance the carry on contamination, that will go to the higher level but not at the lower level. The second thing, ensure that only one dose container is open. So when you are going with the many formulation in the room, so you should not open all the formulation at a time. You have to open the one formulation container and those complete that task, and then only you go for the next dose container. All cases of one group should be identified before the group is dosed. A new catheter or syringe should be used for each dose level. The used container catheter syringe are removed from the dosing station before the new group is dosed. Only one case of animal is open at a time. If it is individually house case, then only every case should be bring and keep back after dosing the animal. If animals house more than one animal in single cages, then after dosing, the dosed animal should be shipped to the another cage. Should not be kept in the same cage, because there are chances are that the same animal may be dosed twice, or one animal may be remain undosed. So it's advisable whenever you are using housing more than one animal in the single cage, then you have to use another cage to transfer the animal after dosing. Each animal should be identified properly by tail tattooing or a microchip or any other marking. And as per case number, so the proper animal will be dosed. The dose volume also should be calculated and keep ready according to the body weight before you go to the rooms, based on the earlier date body weight of the animals. So proper dose of a dose being administered to the specific animal, and you have to record all the activity. So these were the precaution you are required to take while dosing the animals. And for GLP is reference study recommendation, you can use these are handbook, blue, red, and green handbook for your knowledge. This is just for reference. Thank you. I would happy to take a question.
Thank you, sir.
Yeah.
Thank you, sir, for touching on the topic, the test facilities, system facilities in vitro and in vivo in so thoroughly manner.
Thank
Sir,
you.
if
I
your
hope
permission is there, yes, sir.
I hope I could cover all the required information desired by a group.
Yes, sir. Yes, sir. It was covered in detail, sir. In detail, it was covered.
Thank you.
Sir, if you permit, then I would like to ask some questions from the participants.
It's sure. It's my privilege to take a question.
Okay, sir. Sir, Vipul Prajapati, sir, has one question. If we
Yeah.
want to work on viral formulation,
Yeah.
then which type of facilities are required in laboratory?
Viral formulation, see, the formulation is not loaded with the viral. You are talking about the viral antiviral drug or a live virus. That's a
Yes, it
critical.
may be antiviral formulation. It may be
Yeah.
antiviral formulation.
So antiviral formulation is like a drug, what other drug we are using or handling. But if those drug having any carcinogenic effect or any hormonal effect, that's suppose the drug which may have a effect when you inhale or if the particle goes to your body may create some imbalance in your endogenous hormone or enzyme level or having a carcinogenicity effect. Those compound you have to handle in a specific environment that may be a BSL 2, 3, or a 4 level. But for a specific viral compound, there is no recommendation for that. But whenever you want to handle the live virus, and that having effect on the animal or a human body. So in that case, you have to follow the biosafety level guideline. That may be a BSL 2 in case of rabies, but there are many dangerous virus where you have to go for a BSL 4 as well.
Yes, sir.
Yes.
Okay, sir. One more question is there from Guru Prasad. He's asking, isn't BSL 4 labs
Hmm.
the best for in vitro test?
BSL 4 is always good for the in vitro test, but the cost involved for creating BSL 4 is so high, so it is not advisable to create BSL 4 for all the in vitro testing. But wherever it is required and recommended, that test required to be conduct in the such a higher biosafety level, those study or an in vitro test should be carried out in such facility. Otherwise, maximum in vitro test you can conduct in BSL 1, 2, or highly 3.
Okay, okay, sir. Dr. Javed Ahmed Khan is asking
Yeah.
that does GLP compliance in animal test system also work on RRR reduction as
GL,
CPSC has recently emphasized on RRR?
CPCSA and any animal welfare body are emphasizing on R3, or now it's coming R4 as well. But GLP doesn't have any specific thing on this aspect on their agenda, because they are recommend the GLP authority as recommending for a conduct of experiment in a desired manner with expected result, which can be compared internationally. So they are not focusing on definitely they are concerned about the animal welfare, animal health, and some ill animal should not be used, but they are not still emphasizing that if you are going with any animal model experiment, though they will not ask that you go first for a in vitro test and then come, because still the GLP has not specific rules and regulation. But definitely they will promote or appreciate if you use in vitro test system wherever possible in place of in vivo system.
Yes, sir. Sir, if you have time, then two, three more questions are there.
Oh, go ahead, no problem.
Okay, okay. So, Dr. Suvendu Kumar Sahu is asking that in case of prepared, ready-to-use formulations as a test item, can the sample from each batch be retained for a long-term study?
Can you repeat the question, please?
Yes, sir. In case of prepared, ready-to-use formulations as a
Hmm?
test item,
Hmm?
can the sample from each batch be retained for a long-term study?
Each PFS, you are talking about the pre-filled syringe or formulation?
Formulation,
No,
ready-to-use
see,
formulations.
there are different recommendation and guidance for that. If your formulation is stable for a certain time, and then in that cases, you need not. to retain all the sample, but when your stability is questionable, then in those cases you'll have to retain the sample.
Okay, sir. One more question from Rohit Goyal. Is there any specifics about GLP for trading of experimental animals? Trading of experimental animals.
Trading of experimental, as wisely and detail explained, that supplier qualification. There is no specific guidance under GLP, but they have recommended specific criteria for the supplier. So supplier should be like this category. They should be maintain animal in such a manner that the animal should not fall ill, or they should not carry any background infection, or they should be following the PHALASA. There is a one guideline for health monitoring of animal internationally acceptable, that is a PHALASA guideline. So they have to monitor the animal at regular frequency for the all the there is a panel of virus or bacteria that require to be screened every year. Some bacteria virus require to be screened every quarterly, some by 6 monthly, some are annually, and those animals which you are getting from such a supplier should be screened on the desired or recommended panel at recommended regular interval, and it should be free from those infection.
Okay, sir. The next question is from Vipul Prajapati, sir. Is there any guidance or guideline for GLP facilities in relation to the COVID-19?
COVID is very recent disease, and there is no specific guideline, and the guideline which are publicly available is from government. Every 15 days or 1 month they are revising the guidance. But as far as the GLP is concerned, and for a COVID, whatever knowledge so far available nationally, internationally, and on net or a Google, and that you might be aware about the COVID transmission from person to person, or mass to mass, or animal to person, person to animals. There are many questions still unanswered. So it's difficult for any regulatory body to draft the guideline for a disease. It will be definitely available once every aspect of that virus is being studied properly and evaluated. So, but however, with present available knowledge, they have defined guidelines of wear a mask or how to handle animals. There are cases reported internationally. There are dog, tiger, even cat having those who are accompanying to the COVID patient. Such virus has been transmitted to those such animal as well. So there are still many things are going up, research is going on. So definitely there will be a specific guidance, but at present, whatever guidance is available, that is for how you have to protect yourself from normal or a disease or suspected human as well as animal.
Yes, sir. One question from Iswar Tony. Is there any specific guidelines for annual reviews of equipment like CO2 chamber that can be used in euthanasia of animals?
CO2 chamber, euthanasia animal, the chamber is not required to evaluate. It should be neatly clean. But there is a CO2 regulator, the pressure, the volume of dispensing the CO2 to that chamber, and this bottle which the CO2 is filled. So the regulator and monitor are required to be calibrated whether the dial shows the 20 liter per hours or a 10 liter per hours. It is actually dispensing that much volume of CO2 to the chamber or not. So that meter is required to be calibrated at regular frequency, not a chamber.
Okay, thank you, sir, for enlightening us with us the knowledge, sir. Sanjeev Acharya, sir.
Yes.
Sanjeev Acharya, sir. Yes, sir. If you have any questions.
No, I don't have sir questions. I am very thankful, sir, that you covered a very important topic, test system, because this topic is like a day and night you are using this animal experiments in your day-to-day lab experiments, and sometimes we are not aware that there are so many errors we are doing at using them. So this is a very useful session to us, and I request sir that if you can share the slides in a PDF form or any form, so
Sure,
that
sure.
we can share to our participant, because in this FDP sir we have also quiz and examination. They have to secure 60 marks, 60% out of it. So all the participants has to read your slides and have to appear for the exam also. And
Sure.
generally we take the question in the same day.
Yeah.
So if you can email on my email ID, I will
Sure.
share with the participants.
Sure, sure, definitely.
Okay. Thank you very much, Satish bhai. Thank you. This is a
Thank
wonderful session, and it was really long. I think you have taken almost 1 hour and 15 minutes. So really, it was so exhaustively being covered entire points of testing.
Yeah, yes. Thank you. Thank you
Thank
for
you. Thank
all
you.
audience and participant, and
Thank you,
thank
sir.
you for your
It was a
patience.
wonderful
Thank you, sir.
experience. Wonderful
Yeah.
experience.
Thank you.
Okay, sir. Chalo,
Okay.
thank you.
Bye bye.
Really, yeah, thank you.
Okay, all the participants, we will see you tomorrow, and the links are provided in your email as well as in WhatsApp groups. So thank you all. Have a good day.